Hey guys, in this video tutorial we'll be learning about nucleic acid. The nucleic acids are DNA and RNA. These are the principal informational molecules in the cell present in both eukaryotic and prokaryotic. The viral genetic material are mostly RNA. Otherwise, the eukaryotes and prokaryotes they contain DNA as their genetic material. Why DNA is the genetic material? Because it has more stability than RNA. That we will see later why its stability is more. The DNA is also known as deoxynucleic acid, deoxyribonucleic acid. Why it is called deoxy? Because at the 2' end of the ribose sugar, again I'm saying at the 2' end of the ribose sugar, an oxygen atom is absent. This oxygen atom is absent, so it is known as deoxyribonucleic acid or 2' deoxyribonucleic acid. The DNA are formed of monomer called as nucleotides. Suppose this is a DNA. If I will zoom this will form of N. These are nucleotides. A typical nucleotide structure has 3 components. Like one is phosphate group, second is a ribose sugar, and third is a nitrogen bases that attach to 1' carbon. Why the name of the carbon is 1'? Because already the nitrogen bases have carbon and a ring structure. They are already named as 1, 2, 3, 4. To solve this issue, these are named as 1', 2', 3'. Okay. In the sugar, to the first prime, the nitrogen bases are attached. At the 2', the oxygen is removed. At the 3' end, the phosphodiester bond is formed that link to another nucleotide, and this bond is phosphodiester. And at the 5' end, there is phosphate group, and it is the reason why the DNA strand is named as 3' and 5', because as the 3' and the OH group is present and the phosphate and one end phosphate is present, that is known as 5'. Okay. Beside that, RNA is also belonging to nucleic acid. It is also known as ribonucleic acid. It has 3 forms: messenger RNA, ribosomal RNA, and tRNA. The messenger RNA is mostly act as the informational molecule in our cell that codes for protein in the process of translation. The ribosomal RNA help in protein synthesis, and the tRNA help in the detection of amino acid according to the nucleotide sequence that we will learn in future classes. Okay, the nucleic acids composed of monomers known as nucleotides. These are of 2 types. One is purines and another is pyrimidines. So what are purines? Those 2 ringed structure molecule present in DNA are known as purine. You can remember these purines as AG. One is adenine and another is guanosine. First we will go through the structure of adenine. Adenine is also known as 6-amino purine. In the 6th position of the purine, if an amino group is present, then it can be said as adenine or 6-amino purine. So what are the structural detail of adenine? The nitrogen atom, this is the important in the CSIR point of view. The nitrogen atom is present in 1, 3, 7 and 9 position. The naming of the purine starting from here 1, 2, 3, 4, 5, 6, 7, 8, 9 as the functional group, and double bond is attached to here. Hence, according to the rules of nomenclature, it is known as first position, and the 1, 3, 7, 9 position contain nitrogen. The 2, 6, 7 position contain double bond. For the shortcut, I have used this. The 2, 6, 7, that is the beginning of 2, it contain double bond. 6 here double bond, and 7 it contain double bond. And in the sixth position, the amino group is present. And the most important thing is the hydrogen bond. As we all know, this adenine form hydrogen bond with thymine, and in between them 2 hydrogen bond is formed, and these 2 hydrogen bond is formed between adenine and thymine by 1 and 6 position containing the atom. Like here in the 1 position nitrogen is present, and the 6th position our amino group is present. So the hydrogen bond with T is formed via this. This is all about purine. Adenine. Let's see the structure of guanine. This is the typical structure of guanine. It is also known as 2-amino 6-oxy purine. Why? In the second position the amino group is attached, and in the 6th position the oxy group is attached. As the A amino, the primary letter is A, that comes first, that's why the nomenclature is like this: 2-amino that will become first, and then 6-oxy. Okay, 2-amino 6-oxy purine, or also known as guanine. It is also mostly similar to the structure of adenine. It has 1379 position containing N, and like that 267 position containing double bond, beginning of 2, beginning of 6, beginning of 7, and another thing I forgot to mention, also 4, beginning of 4. At this case also beginning of 4. I forgot to mention, sorry for the inconvenience. At the 2647 position the double bonds are there. Beside this, the H bond, as we all know, the between this G, guanine, and C, there is 3 hydrogen bond. There is formation of 3 hydrogen bond in the complete DNA strand. So these hydrogen bonds have formed or attached to the 612 position of this guanine, like this position, 61 this position, and 2 this position, and there will be GC cytosine. Your G will form hydrogen bond with C. Okay, at the position, okay, this is a typical structure of deoxy nucleotide, and there will be only H, not OH. The OH is absent here. So in the first position, in the first carbon, first position 1 prime, the nitrogen basis is present, and this bond is formed between 1 prime and 9 nitrogen position. That is, the position to which attached this bond is attached is N9 of the purine, and in the 5' end the phosphate group is present. Okay. Another is pyrimidines. We can certainly memorize it as CUT or COT, cytosine, uracil and thymine. Cytosine is also known as 4-amino 2-oxy pyrimidine. In the beginning of 3 and 5 there is double bond, and in the 1 and 3 position there is nitrogen, and to the 4th position there is amino group, and 2nd position there is a oxy group. Okay, and the hydrogen bond is formed at 2, 3 and 4 position, that is this and this, and as we all know, C form hydrogen bond with G. Yes, this will form hydrogen bond with G. Another is thymine. In thymine there is 2 oxy group at 2 and 4 position, and a methyl group is attached to 5 position. That's why it is called as 5-methyl 2,4-dioxy pyrimidine. Only at the 5th position there is a double bond. Okay, and 1-3 position there is nitrogen, 2-4 position there is 2 oxygen, at the 5 position there is a methyl group attached, and the hydrogen bond is formed between 3rd and 4th position, that is this nitrogen, 3rd nitrogen and 4th oxygen, and this T form bond with A, 2 hydrogen bond. Another is uracil. This uracil is present in RNA. So we can link it with oxy, that is the sugar. The sugar containing this uracil will be in oxy form, that is in the 3' position there will be complete OH. Okay, this uracil is also known as 2,4-dioxy pyrimidine, and this can be formed by deamination of cytosine, because if the amino group is removed, then it will form a oxygen atom here and form uracil. Like thymine, it has also the double bond at 5 position, and 1-3 position contain the nitrogen atom, 2-4 position has oxygen, and the hydrogen bond is present in 3rd and 4th, that is this position and this position, and it form bond with adenine in RNA only. Okay, let's see the structure of nucleotide present in RNA. In the RNA there is 2' group that contain complete OH group, so it is known as ribonucleic acid, and in both DNA and RNA there will be primarily 3 phosphate, so it is known as nucleotide triphosphate, NTP, nucleotide triphosphate. This can be adenine, guanine, cytosine. Okay, these are alpha, beta and gamma phosphate. At the time of DNA formation, these 2 phosphate group are removed as PPI, inorganic pyrophosphate, plus our NMP. What is NMP? Nucleotide monophosphate. And another thing, at the 3' position, at the 3' position, our phosphodiester bond is formed. Let me tell you about one thing. Ester bond is R C O O R. This can be any alkyl group. Okay. In the case of nucleotide, there is 2 ester. If you will go through this side, this will be ester bond and this will be ester bond. There is 2 ester bond, so it is known as diester bond, and the bone of this ester is phosphate, so this is known as phosphodiester, and it is formed between a 5' phosphate of one sugar and a 3' phosphate of another sugar, and it is the reason we named the different end of the DNA as 3' and 5'. Thank you for watching.